Hello and welcome to week 13 of LET 2150. This week we're going to cover chapter 13 in the book Raymond E. Foster, Police Technology. This week's lecture is going to be the last video lecture for the class, so we'll get right into it. Again, chapter 13 is on technology and investigations. We'll move through the first couple slides here. Assignments, again, this assignment's posted on Blackboard in the assignments section under week 13. Discussion boards, symbols for the discussion boards. There's one discussion board this week. Let's go on to learning objectives here. How technology has changed crime scene protection and evidence gathering, basic science of DNA, fingerprint technology, issues surrounding the DNA databases, digital photography, and investigative software. So we'll get right into it. Forensic science. The word forensics comes from the Latin word foreign. Essentially means to debate. So in modern terms, it has become to mean bringing any kind of science into the courtroom. Any science or specialty brought into the courtroom, such as forensic pathology and forensic accounting. So again, the Latin word for foreign, forensic, is its derivative. Transfer theory. Whenever two objects come into contact, they exchange evidence that they contacted. So that's important to understand that really at the basis of a lot of this stuff, whether it be DNA, fingerprint, anything, transfer theory is at the root of that, meaning that when objects come into contact, when you touch a surface, well, you know, your hair and the cells in your body are constantly being replaced. You leave those behind. There's evidence that you have been almost anywhere. So among those things that we need to talk about, chain of custody is probably one that if you get into law enforcement, you'll get ingrained into your head, quite often. Testimony and documentation that details the location and the condition of evidence from seizure to trial. So that is what the chain of custody is. It assures the court that the items have been entered into evidence are in the same condition as when they were seized. The chain of custody and the procedures used to analyze evidence are more often challenged than the science behind the analysis. So whether or not a documented chain of custody was kept intact, how the evidence were actually obtained, if they were obtained properly without contamination. So those are things it's quite often that the chain of custody is really what is attacked when the validity of evidence is challenged. The Daubert Test. In order for the analysis of evidence to be used against a defendant, the court must accept the scientific conclusion as valid. So the standard for that conclusion is a Daubert test, and it was from a lawsuit, Daubert versus Merrill Dow Pharmaceutical. And what that test concluded is that one is the science derived by the scientific method. It can't be something that's not performed in that fashion and just kind of disguised or covered up by that. Has the science been subjected to peer review or publication? Does the relevant scientific community generally accept the science? What is the error rate of the science technique used to gain information? And are those standards for controlling the technology used to obtain a scientific evidence? These are the standards that the Daubert test puts out there, and again, those are applied to any new science that's brought into the courtroom. It's kind of important that you at least know where this information's at. Crime scenes. A crime scene can also, it's not only the scene at which where a crime occurred, but anywhere that crime or evidence of a crime can be located. So a crime scene is any location where evidence of a crime may be located. For instance, a search of the offender's home may yield evidence, but it's not the location where the crime occurred. And quite often that's going to be the case. They'll go back and search a suspect's home, and that is a lot of times to help derive motive, tie evidence that was collected at the scene back to the suspect. There are protocols for scene protection. Documentation, evidence recovery are critical at all crime scenes. If things are collected by a certain evidence recovery procedure, certain protocols are taken into place to secure the scenes, make sure that the evidence at those scenes are preserved. And quite often, a lot of departments and agencies are now taking the crime scene stuff on the road. This is showing a mobile crime scene van on the top. The lower picture is the inside of that van, just showing it's essentially a mobile lab. So and it's really important because the investigators often refer to the golden hour as the time when the evidence is best recovered, and that's typically the hour just after the crime has been identified. Fingerprint advances. The recovery of latent or hidden fingerprints is no longer restricted to powder, super glue, fuming, laser applications, unique powders. This technology, is advancing, and you can see right here in the picture on the left that this is actually a fuming wand that is taken to scenes. So instead of bringing the evidence back, putting it in a hood and using super glue to raise those prints or identify those prints, they can actually be done on the scene now. DNA basics. This section of DNA will get a bit cumbersome, so I'm sure that some of you have seen these in either a biology class, some of the science classes that you may have taken. But transfer theory demonstrates that wherever we are, we leave behind evidence that we were there. Perhaps our fingertips, but more importantly, our bodies are constantly shedding cells, whether that be hair, skin, those kinds of cells are constantly being left behind. So the collection of those cells or that evidence is at the root of where we do our DNA testing. Body fluids is another one. So it's one of those things that it's where it's that root behind, or the transfer theory demonstrates that that we're going to leave stuff behind. DNA basics. Our bodies contain over 100 trillion cells. Most cells contain a centerpiece called a nucleus. The plural form of that, the nuclei of our cells, contains 23 pairs of chromosomes, which are the biological instructions of who we are. And then those chromosomes, during the conception phase, our parents each contribute half of those pairs of chromosomes, X and Y chromosomes. Inside each chromosome, there are as many as 100,000 pairs of genes. And those genes, the fundamental building blocks of those genes, they are the fundamental building blocks of our traits, hair color, eye color, height, weight, all those kinds of traits. Those are the building blocks for that hereditary information. There are many variations of genes, but generally two classifications. There is a general classification, those that identify us as humans, and the specific, those that give us individual characteristics: blonde hair, blue eyes, height, weight, earlobe, attached earlobe, inner belly buttons that are inner, outer belly buttons. All those traits are in the specific range. Our genes are made up of deoxyribonucleic acid, DNA. DNA consists of a long string of 4 repeating nucleotides. The 4 nucleotides are, here on the left, adenine, cytosine, guanine, and thymine. And they are typically, those bases are paired up. Adenine with thymine and cytosine with guanine. and they create these long repeating strands. So if you look at this, we're going from larger to smaller on this slide. You have your cells. The center part of the nucleus of the cells are where the chromosomes, the 23 pairs of chromosomes are. Those chromosomes then, in each chromosome is where the makeup of the different thousands of genes, and those genes are made up of the DNA in the 4 base pairs of adenine, cytosine, guanine, thymine. So hope that makes sense. The order of the 4 nucleotides, adenine, cytosine, guanine, thymine, with the polymer determine the information contained therein. So, different ordering of the pairs will determine the different information that is kept in that particular DNA strand. And so you can see that the shape of the strand is a double helix. It's a spiral type shape. The complete DNA molecule has a polymer strand with 4 bases. So you can see here what I was mentioning earlier, that the base pairs that group together are going to be guanine and cytosine, and then adenine and thymine. DNA is like digital information, or like digital information has beginning codes and ending codes. They tell us where packets of genetic data or the segments end. With DNA, at the end and the beginning of the DNA strands are the base pairs adenine, thymine, and guanine, cytosine. Excuse me. Most of our DNA is exactly the same, only a small percentage is different from each other. The location where the specific DNA information is located is called the locus. The information that is different between individuals is called the polymorphism. That part examines during that is the part that we examine during forensic DNA analysis. DNA analysis can compare there are 2 different, areas that are ways that they're compared. Short repeating patterns, and look at the variation in length, called variable number of tandem repeats, or the VTNR. The other one that's I think is more commonly used is base analysis of the base pairs, called polymerase chain reaction, or PCR, with polymorphism on specific genes. So recall in general and specific. So general genes, DNA, are called monomorphisms, and the specific gene DNA is called polymorphism. So DNA analysis is conducted on the polymorphisms. There's the specific genes DNA. Like fingerprint information, DNA is information that can be converted to a numerical value for ease of search. Combined DNA information system, CODIS. I'm sure that watching shows and whatever you talk about CODIS database. And actually, the CODIS database is a combination of databases. There are 153 laboratories in 49 states that are a part of the CODIS system. It actually includes 3 different types of databases: convicted sexual offender, other offenders, and missing persons. Those are the combination of the CODIS database. Some of the concerns with the database, and these are all questions that are debated quite often, and quite often debated in the courts. Who is included? Who can be included in the database? Convicted, arrested, or suspects, and that varies from state to state. What about juvenile? Juvenile arrest records do not necessarily follow the juvenile into adulthood, but what about DNA that's collected and put into that database? Does it? Since a portion of DNA information is familial or family related, if your sibling or parent is in the database, so part of you is. If you are a familial match for DNA, so there'll be some match there. And what about research? How is some of that information included into these databases? And that really leads us into today's or this week's discussion board question. So based on your knowledge of DNA, go to the discussion board and post your response to the following. Does the government have the right to collect DNA from criminal offenders for use in future criminal investigations? What are the areas of greatest concerns? Do you agree on why or not? Why or why not? Good question. Oops. Now let's talk about some of the softwares that are used in investigative work. So one of them, and most common in most departments, typically have some way, shape, or form of this, is a case management software. Determining how cases are assigned, who's assigned, what workloads assigned, types of cases, coordination and productivity, closure rates of cases, and that's often used by police administration to evaluate the performance of their investigators. Also, crime scene drawing software. Crime scenes are often used as a mean of drawings are often used as a mean of documenting the location of evidence, assisting in determining the sequence of events, and so forth. So, crime scene drawing software. And then the other one that I think is kind of neat is the link analysis software. TV shows that you watch, you'll see them putting up whiteboards and they're listing the subject and pictures and that, trying to draw the links or the connection between victim and suspect. Timelines are often a part of that link analysis software. So it's another good feature. So this software really replaces the big whiteboards where information is kept up and often referred to as their crime board. Digital photography is a picture stored in language. Okay, it's a digital picture. I'm sure we're all aware of what those are. One thing that's important is that digital imagery is more easy to manipulate than traditional photography. So, additional software and security procedures and protocols are necessary to maintain the chain of custody. So, to kind of alleviate the question of whether or not a photo has been manipulated or changed. Another thing that we see and more so in crime or crash scene investigation is the use of high definition survey. It's a laser technology that gives you a 3D representation of a crime scene or a crash scene. And then this is a photograph provided by that. This is a three-dimensional representation of a man seated with a gun. So it gives people a way to reconstruct a crime scene and to go back and look at it at a later date. So that's chapter 13. That is the last chapter in the book that we'll touch upon here. So if you have any questions about where we're at here, remember this week this is the last week. Next week, I hope everybody has a nice week off. Enjoy the holiday, Thanksgiving holiday. The week after that, we come back and we're going to do some review, and then we'll hop right into finals week. So we're at the end here. So hope everybody has had a good time so far this year in class. If you have any questions, please let me know, and we'll get them addressed. And thanks, guys. Have a good week.